Okay, good morning to everyone. And here is our final assignment from Group 2. And the title for the assignment is the gut-brain axis in Parkinson disease pathogenesis. And here we actually dig into the theoretical background of Parkinson disease and the causes to it, specifically because it is actually defined as a systemic disease, which is caused by the degeneration of dopaminergic neurons in the brain. However, some of the symptoms that we all know of Parkinson disease are, of course, physical symptoms like, for example, tremors, stiffness in the muscles, and the slowness of movements over time. However, also to date and so far, this disease has not a treatment, a specific treatment that can actually solve the problem, but it can slow down the symptoms, which are the ones that we found as specifically as motor symptoms. And here, the levodopa treatment is one of the most common standard medication used for treating this disease. And from this interest in this disease, we actually found more in depth which could be some of the causes. And here we found some studies that actually show that the gut microbiota, so all the bacteria and that stays in the gut, are actually one of the essential components, and they play an essential roles in the neurological development of this disease. Indeed, of course, as you can see in the picture on the left, for example, here we could have an inflammatory trigger that could be like toxins or antibiotics that can initiate an inflammatory response in the gut and cause a bad gut microbiota. So this bad gut microbiota actually, and the inflammatory and the inflammation that develops in the bowels actually can cause an increase of the aggregation of a protein, which is the alpha-synuclein protein in the gut. And this protein, the aggregation of this protein, is actually one of the main manifestation of Parkinson disease in patients, which through the vagus nerve can be transmitted from the gut to the brain, so from the enteric nervous system to the central nervous systems, and in that sense supporting this hypothesis that the gut microbiota may play a pivotal role in the pathogenesis of Parkinson disease. And from here, indeed, the intestinal inflammation may initiate a widespread systematic inflammation and synucleinopathy in the human body, which together actually drive to the neural inflammation and neural degenerations, which of course translates into the loss of dopamine synthesizing neurons, which then affect motor activations and causes the progression, of course, of the disease and the symptoms that we are used to know. So this was our background, and from here, one of the main research questions that we developed is to try to understand how does this bad gut microbiota may influence the mechanisms of Parkinson disease in patients that are affected by this disease. And so, as you could infer from before, the theoretical background actually it rests on this gut-brain axis, and so the connection that there could be between the gut microbiota and actually the loss of dopamine neurons in the brain. So, of course, the relationship between the enteric neural systems and the central neural systems. And for developing this topic, and to answer actually these research questions, we develop a simulation models, which is an adaptive network model with second orders of model levels. And we used as the main functions, the AI logistics and step mode functions. And here you can see our conceptual models, which at the center you can find actually the connection between, on the left part, the gut processes, and on the right part, the mental processes. So as you can see, for example, in the left part, some of the keywords that I used before, like the bad gut microbiota and the inflammatory trigger intestinal inflammations. On the other side, on the right part, there are all the systemic inflammation, neural inflammation, and then neurodegeneration, which are related to the brain part. So, and this synucleinopathy, which are the increase of aggregation of the alpha-synuclein protein, both in the gut and in the brain, which are actually, which actually influences themselves in a loop. So this, and I leave the floor to Catherine.
Yeah, so here we present you the adaptive network model that we created, and you can see that on the base level, we redemonstrated a bit what Korinzia just came to explain. And so what we added in red arrows are negative influences that are going from states. So we can see that the constipation has a negative impact on the bad gut, but also the alpha-syn in the gut has a negative impact on the bad gut. Then we have negative impacts from the degeneration towards the dopamine levels in the brain, and the same occurs for the dopamine levels negatively affects the degeneration. So in order for this model to make sense, because we have actually a lot of different time points that we are considering here. So we have some neuroinflammation or degeneration that is taking place, and this is a process that takes years to occur. And then we have here probiotic intake, constipation. That's something that can occur on a daily basis or over weeks or months. And therefore we created in a second level weights. So here we have the weight for the bad gut and the intestinal inflammation. And we did this because the bad gut makes you more sensible for intestinal inflammation, and thus strengthens the link between the inflammatory trigger and the intestinal inflammation. We also have here then introduced in a third level the speed learning that makes it how fast this weight will impact that connection. Then we created a weight for the dopamine level and the motor activation. And here we have the dopamine levels that make you more sensible for motor activation, because when you have a lot of dopamine, you work better and your activation is better than when they are low. And thus this strengthens the link between the stimulus for activation and the motor activation, which you can see here. Again, we added a speed learning that will influence this weight. Then for the levo, which is the levodopa medication, here we have the intake and the effectiveness of this medication. We added a threshold. This threshold is influenced by the levodopa that you take in, and this threshold has an impact of the effectiveness, because even though you can increase the intake of the medication, the effectiveness of it will reduce and weaken over the time period. And again, we created on a third level a learning speed on how fast this effectiveness will weaken down. And so we did the step mode functions for probiotic levels, fiber levels, stimulus, and levodopa intake. So these will occur at certain time points during the simulation. For the rest, we use the allogistic functions, as Korinzia already told you before. So in this graph, you can see this very simulation of the active network model. On the right-hand side, we gave you the legend with all the different states and the colors. But we can actually see that we demonstrated our hypothesis with the alpha-synuclein that is increasing in the gut through bad inflammatory triggers, a bad gut microbiota, and then we have increased our activation of the motor activation will decrease over time, which is our outcome. What happens in between, we can see that through our threshold that we created for the levodopa intake, which will get more sensible over time, we actually decrease the effectiveness of the levodopa medication over time. We can also see that all the inflammations that we have in our system are constantly increasing, and the downward is actually every time we actually take some good probiotics, or we have high levels of probiotics, or we have high level of fibers, we have a small downgoing, but once we actually don't have this anymore, it is going back up, and it's a gradual increase. We can also highlight in this that there is a steady degeneration happening within our patients during the time. And so we actually managed to represent a model that is demonstrating the biological as well as environmental factors in Parkinson's disease. What is the impact for future work? So we actually used a more simplified manner. So we looked at the gut microbiota, or like the bat gut microbiota composition, and the brain as itself, and how they influence their mechanisms. And we took into account the medication and external factors here, the toxins, as well as fiber and probiotic level intake. However, for future studies, there should be more specific looks on how these specific probiotics can influence the action, and also at other functionings, such as the mitochondria, that play also an essential role in the Parkinson's disease, to have a more complete overview.